Thanks, Matt. I really appreciate organizing this colloquium. I've learned something already, and I look forward to learning more, and maybe I'll even contribute a little bit. I think this perennial agriculture issue is very important. I do my research in agricultural areas. I do my in western Minnesota, corn and soybean land, and my study site is very hilly, and there's we could really use some perennial agriculture. But I don't do agricultural research. I'm a conservation scientist, and I study native prairie plants, the ecology and evolution of long-lived perennials growing at the margins of corn and soybean fields. So what I thought I'd do today is just tell you a little bit about the project that I've been working on, and hopefully I'll highlight some issues that I think might be relevant to perennial agriculture, but you will probably be able to judge better for yourself. So the Echinacea project, it takes I started it in 1995 when I was in grad school. Use a model organism approach to study ecology and evolution in fragmented prairie. We've had funding from NSF for a very long time, which is great. They're interested in long-lived plants. I'd like to start off by acknowledging this is a big team effort. I have lots of great folks to work with. And let's see, Amy Wananen is here. She's Amy Insink Wananen. She's going to give her poster presentation this evening. I encourage you to go. So the narrow-leaf purple coneflower Echinacea angustifolia is what I picked as a model organism because it's typical of a lot of prairie plants. It's in the Asteraceae, an important family. It's long-lived. I did not know how long-lived it was when I started. In 1995, I tagged a few hundred plants, and back in last year, 2017, a lot of those plants were still alive. I had no idea what I was getting myself into. They're strongly taprooted, and they reproduce via seed. They're heavy and dense, don't disperse. Pollinated by generalist native solitary bees. And, it's strongly self-incompatible. It's got a strict sporophytic system so that a plant like this can't self-pollinate, and also it will reject pollen from other plants that share S alleles. So our approach is to look at both ecological and genetic processes at the same time and how they influence population persistence. Just to go through the talk, I will organize it by looking at the life cycle of the plant, and then that'll kind of break down, and then I'll do a little synthesis. So we'll start off with seeds, and really the fruits of the purple coneflower, kind of like the shells of sunflower seeds, they're big and they're hard, and they form regardless of whether the plant was pollinated. Those X-ray images on the top show that sometimes they're very well pollinated, and sometimes they're not pollinated at all, like the bottom image shows. They're totally empty. But if you look at them from the naked eye, you can't tell. So and we have no evidence that this seed set is related to resource limitation at all. It seems to be totally driven by pollination. Okay, so the next step is the transition to seedlings. We used to grow lots of plants in plugs, and it's very nice for us. It's convenient, it's air conditioned, and it's very nice for the plants. Now in nature, life is very different. So we've taken to studying throwing out seeds in the fall like would happen in nature and see what happens. And it's complicated. Every year is different. And I did a study in 3 years where we put out seeds in the fall. And to cut to the chase, emergence per viable seed was best in 1 year, and I'm showing that year right here. We had 43%. Burning played a big role. If there was a fire beforehand, we had high emergence, and if there were burns, no burns, or burns afterwards, germination was very low. Our emergence was very low. And also the type of vegetation. Old fields were mostly better than restorations. So environment plays a big role, but what about the genetic basis for emergence? This is a question we'd really like to get at, and we have an experiment in the ground. I'll let you know in a couple of years how that works out. Yeah, throwing seeds out in the fall and pedigreed seeds, it's nerve-wracking. So we've looked at seedlings out in nature, and it takes a big effort to find them. We did it for 7 years, and that was very trying, but we found 955 seedlings, and we followed them every year since. During the summer, they seem to survive pretty well, from 55 to about 80%. So that's not very interesting. And then we still have over 10% still living as of last year. So that's the seedling stage. They can stay seedlings for, or they can stay juveniles for quite a while. So the juvenile stage is also driven a lot by environmental things that happen in nature, like fires. We'll come back to this again and again. Fires in old field type vegetation are not so good for juvenile survival, but it's not too bad. However, fires are critical to the survival of the juvenile stage plants when grown in restoration. And it's probably restorations are so dense that the shade doesn't allow the juveniles to survive. Now this is the next step is, well, how long do plants stay in that juvenile stage? And how long does it take till they first flower? And so we have a couple of experiments that we've been working on to look at that stage. And the first one is the one I just mentioned, when we have these former seedlings that we follow. And of the 955, only one has ever flowered. And it tried to flower, but it died. And then of the 102, they're still out there. So if they all flower, or if they are all flowering right now, then the average time they would be in this juvenile stage would be between 8 and 9 years. Okay, it's going to be longer than that. I don't know how much longer though. This experiment, we did a common garden experiment, and this shows you that there's a lot of variation in the time to first emergence. You'll see that, and these were pampered plants. They started off in containers. And some of them came up when they were 3 years old, a lot when they were 4 and 6 year old. The asterisks are years right after a burn. And then there are some plants that flowered for the first time at age 16, and there are a couple that still haven't flowered yet, age 21. So it's a long, drawn-out process. So let's talk about adults next. And what's interesting about the purple coneflower is it alternates from flowering to non-flowering, and the number of years that they flower in a row is variable, and the number of years that they don't flower in a row can go up to 19 years that we've documented. What influences this crazy behavior? Well, we have some ideas. I'll tell you about one experiment, or one observation that we've done on a nature preserve, Nature Conservancy Prairie Preserve, where there's 2 burn units that they burn approximately every 5 years. The west unit is shown in the dashed lines, the solid line is the east unit, and you can see that the proportion of living plants that flowers is about over 40% every summer after a burn. And it's less than 40% any other summer. So it seems like this burning really induces flowering. I don't know this, but I don't think it has to do with resources. I think it has to do with some kind of signal that may be separate entirely from resources. But there is something related to resources going on, because the number of heads that a plant will produce varies a lot. The mode is one head per year per flowering plant. But you can see that in some years that they produce maybe 2 or on average 3 heads. And that only occurs in years following a spring or fall burn. So what I presented so far is kind of like a hodgepodge, mix match, a bunch of different disconnected studies of different life history stages. We call them components of fitness. And this is the wrong way to go about studying a long-lived plant, because what we really want to know, like say, if we think fire is important and we study old plants, we don't know if the old plants that we're studying, how they dealt with, if you will, when they were young with fire. Maybe they loved it, maybe they hated it. Sorry to anthropomorphize, but we really don't know. So what we need to do is we need to have a better way, and that is Darwinian fitness. We have to look at survival and reproduction together. And that poses some problems. It's a challenge. How do we look at this, the big picture? And the first is that what I just said, fitness components for a given individual is conditional on the individual state for an earlier component of fitness. And then the other thing is, well, if you put it all together and you're going to quantify total fitness, you're going to be counting seeds probably, or fruits, and there's no really good probability distribution that's suitable for modeling all these components together. Well, there is a solution. And that is, well, let me show you one of these nasty distributions. This is an experiment of 7-year-old plants, 1,300 7-year-old plants. This is a distribution of total lifetime fitness. Notice that there are about 1,000 plants that are zeros. They produce 0 seeds by age 7. But notice that some of them, 300, they're stuck at 0, but 700 or so actually have the potential to increase their fitness. There's a whole bunch around 150 and 200, and then there's, one super plant at over 1,000 fruits. How do you analyze that? There's just no way looking at one distribution. But with aster modeling, which was developed by Ruth Shaw and others, Charlie Geyer and Julie Ederson sitting right here, the aster model is a way to tackle this problem. And I'm just going to tell you a little bit about it. It's like a generalized linear model. It's implemented in R, and the response is, instead of just a number, it's a graph. And I'll explain what that's like. So if you imagine the longitudinal life history of a plant on the top, it's like basil. It's living the whole time for 6 years in a row. The second plant lives, and then dies, and then stays dead. That's how it works. And the next one is basil, flowering, basil, flowering. Okay, so how do we account for this? Well, we can make what's called a graph, and we figure out, we start off with one plant in year 1, and we say, does it survive to the next year? Yes, no, and then it dies and it stays dead thereafter. Okay, but then we look at, for those that are living, do they flower? And this we can model as binomial yes or no. And those that are actually flowering, if they have a yes, then they would have a head count, which we can model as Poisson distributed. And if they are actually have produced heads, then they can produce fruits, and which we can count them all out. Like Matt knows all about counting fruits. So we can count till the cows come home. And so this is a way that we can assess lifetime, or at least long-term fitness for plants that have any kind of natural history that we can characterize in graphs. And so I'm going to quickly tell you a couple of experiments where we have used the aster approach. They all come from this one experimental plot in western Minnesota. First, we estimated fitness in 7-year-old plants with standard errors of inbred plants and non-inbred plants. And we can compare them. The difference is inbreeding depression. So what these numbers represent is the inbred plant, given it was planted as a seedling, it would produce, on average, we'd project between 20 and 30 fruits over the course of its 7 years, compared to a non-inbred plant, which would produce over 80. We can also use the aster approach to estimate fitness coarsely, heads per plant, and look at population differentiation, which is important for looking at in isolated prairie remnants. And then we can look at more populations. We can even use continuous predictors with aster models. And then the last result that I'd like to share with you is that the aster approach implements random effects. And we did a big experiment where we developed pedigreed seed to estimate quantitative genetic additive variation of fitness. And lo and behold, we see lots of variation. And this is breeding values for fruit count. And you can see whatever the mean is, there's these super plants out here. So what that is a very helpful message that there's lots of genetic variation and there's the capacity for this population to evolve by natural selection to have increased mean fitness. So the key insights, that there's lots of variation in these measures of fitness. Fire plays a key role. We can quantify long-term fitness. An aster will rise to the challenge, and that there are lots of genetic contributions to variation. Thanks. Question? One question while it goes up.
Is that mass distribution of fitness in a burn plot or under?
One that does have a history of biennial birds, yeah.
Biennial burn.